Ladies and gentlemen, good afternoon. Thank you for coming. My name is Tom Durig. I'm the Pharmaceutical R&D Director for Ashland. And this afternoon, I'd like to present our discussion on role of excipients in solid amorphous dispersions, in the design of solid amorphous dispersions for drug delivery of poorly soluble drugs. First, I'd like to discuss a little bit the topic of why solid dispersions, why amorphous solid dispersions is important. Then some of the design considerations for solid dispersions. Take a look at some of the current manufacturing technologies currently available and commercially useful. And then close with two case studies on rational solid dispersion design. One for solubilization of poorly water-soluble drugs, the other one for controlled release of a poorly soluble drug. So first off, just some general comments about low solubility and amorphous dispersions. Bringing a new molecule to commercialization today has become a lot more challenging for various reasons. And one of the main one in terms of oral drug delivery is the large increase in the number of poorly soluble and poorly bioavailable compounds that we have to deal with. And there's a number of approaches that have been developed to help with this problem. Among them are particle size reduction, things like nanomilling, and you see some examples on the screen like Emend and Repimune as commercial examples. Amorphous solid dispersions being also one technology that is increasingly being used. One can also formulate co-crystals where you form a co-crystal between a drug and another ingredient. And then, of course, lipid-based systems, emulsions, nano-emulsions would be another solution to these poorly soluble, oil-soluble compounds. And cyclodextrins or other kinds of complexes where we form a complex between drug and an excipient or polymeric carrier is another option. Cyclodextrins being a very notable category there. And then, of course, one can also look at modifying solubility with approaches such as salt selection, as well as selecting specific crystal forms, polymorphism. But amongst all of these types of approaches, our topic today will mainly focus on the amorphous solid dispersions, which are clearly an area of burgeoning and growing interest and commercialization in the pharmaceutical industry for this kind of problem. An amorphous dispersion in its simplest might be described as a dispersion at the molecular level or solution at the molecular level of an active ingredient, an API, in some sort of a carrier base. Quite often, the carrier in which it is molecularly dispersed would be a water-soluble polymer. Some of the specific reasons why amorphous dispersions tend to increase solubility is because in an amorphous dispersion, the API will be in a metastable state, an energetically higher state than it would be if it was in crystal form. But when one looks at solid amorphous dispersions, there's an additional reason why these are often selected over other approaches to enhance bioavailability. And it has to do also with permeability, because we have to remember that when you apply a certain solubilization technology to increase bioavailability orally, it's not just that the API has to dissolve in the gastrointestinal fluids, but it actually has to be bioavailable after that and actually permeate across the gastrointestinal mucosa in order to be active. And in this area is where amorphous dispersions often enjoy some significant advantages over some other technologies such as, say, surfactants, co-solvents, and cyclodextrins. So, for instance, if you include a surfactant in a formulation, it will very readily enhance the solubility of the API in the aqueous medium of the gastrointestinal tract, but the surfactant would continue to be have a high affinity for the API and vice versa. So they would tend to be remain associated, which will compete with partitioning into the gastrointestinal mucosa. The same might be true for such things as emulsions and cyclodextrin complex type systems. On the other hand, once an amorphous dispersion is dissolved in the gastrointestinal tract, there's really not much further interaction that would limit partitioning of the API into the mucosal membranes. So how do solid dispersions really solubilize drugs? How does it really work? And we like to think here in terms of the spring and parachute concept. So at its simplest, what you see on the screen here, the blue dots would represent a molecularly dispersed drug, dispersed in a polymeric, suitable polymeric carrier. And if this dispersion is then swallowed and ingested, you should expect that in the stomach it would start to interact with the fluids and dissolve. And the particular benefit we get is that, first of all, because it's an amorphous dispersion in this non-crystalline, higher, energetically higher state, this metastable state, we have a massive boost in solubilization. So this is the spring part over here. So typically, if it was just the crystalline API, in a tablet formulation, the solubility you might get is this lower line here, this dotted red line. That would be the inherent solubility of the drug in the gastrointestinal fluid. But because it's in the amorphous state, you get this very sharp rise here. So this represents the spring. And now at this point, you're many folds higher in concentration than the intrinsic solubility. So this would be regarded as a supersaturated state. And the problem with supersaturated solutions is that they also are unstable, and the drug would naturally tend to want to form little crystals at this high level here in terms of concentration, and start precipitating. So the risk is always, if you do induce this highly supersaturated state, much higher solubility, that you might get very rapid recrystallization, sort of like a sharp little peak, so that you really didn't get much benefit. So the other benefit and the other purpose of doing an amorphous dispersion, if it's done well, is that you might be able to induce what's called this parachute effect. So instead of a rapid precipitization here, a rapid crystallization, what you'll have is a more gradual decrease in concentration over time, so that we actually have a nice long absorption window that might last up to 2 hours. So typically, a well-designed amorphous dispersion would ensure that you're somewhere at about 80% of the maximum concentration that you were able to achieve for up to 2 hours. And in so doing, we can really ensure improved bioavailability. So when it comes to additional design considerations for these kinds of amorphous systems and formulations, there's really three categories we have to think about. There's design considerations related to the API and its properties. There's design considerations related to the type of polymer we're going to choose, which polymer to select. And there's design considerations related to the actual process, the manufacturing process that we would use. So in terms of API properties, there's really, again, three buckets, and you can see them here on the screen. Primarily, there's biopharmaceutical considerations. This would relate to the dose. How large a dose do we have to deliver? Permeability, as we just spoke, in terms of permeating across gastrointestinal mucosa, as well as absorption windows, because perhaps this drug is only absorbed in a limited part of the gastrointestinal tract because of some kind of active transport mechanism, for example. In terms of physical chemical properties, they really are important from the point of view of stability, processability, and being able to interact with the polymer. So we're always interested in indicators of crystallization behavior. So the melting point, the TM here, that's the melting temperature, the glass transition temperature, crystallization temperature, or recrystallization temperature. These are all important parameters to give us an idea about how stable would this amorphous dispersion be. Also, to some extent, you'll see the same terms over here in processability. They help you guide you in terms of if you were going to do hot melt extrusion. Also very important are things like pKa and hydrogen bonding capacity. This would guide you really in terms of what kinds of polymers you might select in order to have a good interaction. In terms of polymer selection, really two main things to focus on. One is what would be the best polymer in terms of assuring desirable interactions with the API, because the polymer very much has to be playing a role of stabilizer, solubilizer, and then also preventing that precipitation once the drug is dissolved in that supersaturated state. So dispersion forces and hydrogen bonding often are some key parameters to look at. Also, in terms of processability, we want to be thinking about glass transition temperature, especially if you were going to trying to do hot melt extrusion. And of course, some of these same properties would also affect polymer solubility, which if you were going to do spray drying is very critical, because you'll need a suitable solvent in the end that can dissolve both the API as well as the polymer. Some of the common polymers used, typical polymeric carriers for solid dispersions, would be the non-ionic or non-enteric polymers such as copovidone, povidone, hypromellose, as well as hydroxypropyl cellulose. And then really the other big category that tends to be finding a lot of use would be the ionic or enteric polymers, among which hypromellose acetate succinate might be the most commonly used one, and then hypromellose phthalate, cellulose acetate phthalate, and also polymethacrylates. So the second category are all ionizable type of polymers, generally ones that don't dissolve too well in an acidic medium. The ones which are the most commonly used, I've highlighted in red. When we think about, what kind of manufacturing processes and technologies would we want to bring to bear, commercially, currently, there's really two processes that are most viable. There are many other ways in which you could conceive of making an amorphous dispersion, and there are one or two methods that are also used commercially, but by far the two most common ones would be hot melt extrusion and then also spray drying. And it's quite interesting in terms of seeing the evolution of these two technologies, because you would have said that until perhaps this year, the majority of marketed compounds that were in the amorphous dispersion state would have been produced by spray drying or some kind of solution precipitation type of process. And hot melt extrusion, we really had one very big current common example, that being Kaletra, ritonavir, lopinavir combinations by hot melt extrusion. But even in the last 6 months, there's actually been quite a lot of change. And just recently, Merck and Company, among others, have gained approval for 2 new hot melt extruded formulations. So the balance between spray drying and hot melt extrusion, I think, is rapidly changing to one where there's several more commercial examples of hot melt extrusion now. They both have their advantages and limitations. Just very quickly, let me hit some of the highlights. So generally, one would envisage and obviously conceive that hot melt extrusion could be a little bit more cost efficient. You're not using special solvents that require all kinds of precautions. You basically, it's all done in one step. It's also a continuous process. The equipment tends to be somewhat simpler. However, some of the limitations are that it's clearly not suitable for all APIs, because by definition, hot melt extrusion will mean that the API needs to be able to withstand some kind of heat treatment. For thermolabile compounds, this can be a challenge. And in some very challenging cases where there may be a really strong tendency to recrystallize also in the solid state, you may not be able to succeed with hot melt extrusion, because inherently, although there's a lot of research being done on this aspect of hot melt extrusion technology, inherently the mixing efficiency in a hot melt extruder tends to be less than it is when you have a drug and a polymer completely dissolved in a mutually acceptable solvent, and both things are in the true solution state. So you don't quite get the same intimate mixing as you do in spray drying. So for spray drying, that's really one of the major advantages. If you truly want a perfectly mixed, intimate interaction between the polymer and drug, spray drying is the state of the art currently. It too is a continuous process, or can be run as a continuous process, one would say. And then, of course, the other big advantage is because you're not worried about will the polymer be thermoplastic enough for extrusion, you tend to have more choices of polymeric carriers in spray drying, whereas those tend to be a bit more limited in hot melt extrusion. But some major issues and costs are there, and these tend to be the limitations. Obviously, the use of organic solvents can, especially on a industrial scale, can be more complex and would tend to add costs in terms of both the kind of equipment one runs and the process, ongoing process verification. So they're really complementary technologies. We tend to see them as not, it's going to be the one or the other, but with these two technologies, it's possible to address the majority of formulation challenges one has in amorphous dispersions. So in terms of case studies, the first case study I'd like to talk about is designing a solid dispersion using HPMCAS for a very strong crystallizer. So the drug involved here is Ezetimibe. This was chosen as, first of all, obviously a very low soluble drug, but one which has the characteristic of being a strong crystal former. So this is a drug where you have both challenges in maintaining the amorphous state in the dosage form, so the solid tablet dosage form, and preventing it from recrystallizing before it is given to the patient, as well as then once it's actually dissolved in the gastrointestinal tract, and you have that, if you remember the spring effect, when you have that high saturated solution, Ezetimibe tends to recrystallize or precipitate very rapidly. So to extend that parachute tends to be a little bit of a challenge. We chose here a drug load of 60%, again, because that's a much higher challenge than low doses, and to really discriminate the effects of the different kinds of HPMCAS that are available. The process used was spray drying, so we made spray dried dispersions here with the Aquasolve HPMCAS grades L, M, and H. These were all used at in the spray dried solution was at a 5% concentration. And then the spray dried dispersions were evaluated afterwards for kinetic solubility as well as stability. Some of the properties of the drug are shown there in the table. So first of all, in terms of stability in the solid state, we tend to run these at accelerated conditions for different lengths of time. So one of the standard tests is crystallinity or ability to withstand recrystallization in an open environment for 65 hours at 40 degrees centigrade, 75% relative humidity. And here you can see DSC thermograms for the three formulations that were tested. So the blue one is the one which contains the HPMCAS L grade. And you can see here after 65 hours under these accelerated conditions, there's a little melting peak here that corresponds to the melting temperature of the API, of Ezetimibe. So there has been some recrystallization. For the M grade, the red line here, you can see that we have two glass transition temperatures indicating phase separation. And you can kind of nicely see this here corresponds well, the 67 corresponds well with the glass transition temperature of pure Ezetimibe, and the 120 degrees is approximately the glass transition temperature for HPMCAS. On the other hand, our grade H, the green line here, so the formulation where we use the H grade, shows that after the time has elapsed, we still have the same amorphous dispersion. There's just one glass transition temperature roughly at the point of the Ezetimibe. So no phase separation. So while there's no recrystallization, this red line also tends to indicate a less stable state, because Once you have phase separation, usually in some subsequent time period thereafter, you should expect recrystallization. So in terms of stabilization effects, we clearly saw that the H grade here tended to be more effective than the M and L grades. And we think hydrophobic interaction is playing a role here. These different grades have different acetal substitutions. The acetyl moiety on the HPMCAS tends to be the one which affects hydrophobicity the most, and H has a higher level. If we looked at the dissolution behavior, we again see some interesting discriminating behavior between these. On the red line here, that would be the HPMCAS H grade, and the green would be the M grade. You kind of get some form of a parachute, as you can see. We're extending a relatively higher concentration for maybe an hour or two. On the other hand, the L grade, which is the blue line here, you see a very sharp peak, so much more effective in terms of a quick high boost in terms of a spring, but very rapid recrystallization, indicating that there's really hardly any interaction between drug and polymer in that solution state, and it just pretty much all precipitated rapidly. Again, we think this has to do with the both the polymer dissolution rate, but also the hydrophobic interaction, with the acetyl groups being more at a higher level in the M and H grades. I think I've said most of everything on this slide, so let's move on to the next case study. The next case study is an interesting one in that we're trying to do two things at once. We are at the same time trying to address a challenging low-soluble drug, so increase solubility, but at the same time control the dissolution rate over a period of up to 8 to 10 or 12 hours. So the challenge here is not only do we want to solubilize, but we want to maintain whatever drug is dissolved without reprecipitation for a long time. The drug chosen here is nifedipine. You see the properties to the right. And then the approach chosen here was extrusion. So we're actually trying to make an extruded pellet by combining copovidone as well as HPMC. So two different polymers. Copovidone was chosen because it's well known as an excellent extrusion polymer, very thermoplastic, good extrusion aid, also used in amorphous dispersions. And then secondly, HPMCAS is known for its excellent ability to maintain that parachute to prevent the recrystallization in solution, but also it's an excellent controlled release polymer. So the theory was if we use appropriate ratios of these two, we might be able to achieve both objectives. So the concept here is a bit different from the hang glider effect, from the spring and parachute effect. Here we have the hang glider effect. So we are both solubilizing and continuing to maintain a dissolution rate over a long period without, at some point having some rapid precipitation, because you could think that as soon as the drug releases, it might precipitate. So in fact, you wouldn't get this nice dissolution curve here, but something much lower, because while the drug dissolves, it might also reprecipitate. And again, the blue line at the bottom would be the natural solubility of the API. So here you see some of the formulation and process factors we used. We varied the concentration of the API between 10 and 50%, and the HPMC was varied between 0 and 40%, and then the copovidone, again, used at different ratios. We also tried some trials with a small amount of polyethylene glycol as a plasticizer here, just in case we had problems with extrusion. And we tried different pellet sizes, 2, 3, and 5 millimeter length. We used a Lystritz 18 millimeter extruder, and here you see a typical temperature profile for our machine. When we looked at the different levels of drug loading, we very quickly realized it wasn't going to be feasible at, say, 50% drug load. As you can see again, there's a very strong melting peak here showing that the formulation had recrystallized. On the other hand, at 20 and 25% drug, there was really no sign of recrystallization. The formulation remained in the amorphous state. when we looked at the effect on dissolution profiles and varying different grades of HPMC, we saw some very nice results. As you can see here, in a formulation with 20% drug, 40% HPMC, and 40% povidone, and then varying the different kinds of HPMC from K4M to K200M, so roughly 400,000 to 1.2 million Daltons. We get a very predictable release. We can pretty much select or program the rate simply by changing the molecular weight of the HPMC. And as you can also tell, there really wasn't a great amount of crystallization or reprecipitation out of solution. We were able to sustain this for a good 8 to 10 hours in some of these examples. It's really only when you reach close to the peak release did we see a gradual decrease. So we were clearly able to achieve both a spring and parachute and a hang glider effect. We're also able to control release with pellets by just modifying the size of the pellets. So you see here release profiles for 3 different sizes of pellets, 3 different surface area to volume ratios. So very direct correlation. And lastly, in terms of dissolution stability, these formulations did remain stable for up to 6 months at accelerated conditions. So really no change in terms of stability. With that, I think we'd like to conclude. Thank you very much for your attention.